All right. So we are going to be doing. I might as well grab this. We're going to be doing a brain dissection from the lovely folks at carolina.com. You can go and you can buy this on your own. I would say it's totally worth it to buy it on your own. Carolina Biological is their full name. And they send you the brain. I'll show it on this because my phone camera is going to be the best out of all of them. Carolina's perfect solution. So sheep brain. And then just in case you get any of their perfect solution, which is proprietary in your eyeballs or on your skin or anything like that. It is not good to be in there. And then, so here it is. I'm going to open it up in a little bit. Okay. And then we'll go through. So it's in this perfect solution. It's a little vacuum sealed, so it's a little smushed. I gotta make sure I gotta stay in the right places for these damn things. So there it is. Looks pretty, doesn't it? I've got my nitrile gloves right here to make sure that I don't get this perfect because it's supposed to be a skin irritant. I've got they give students this knife, and I thought that was hilarious. So I'm going to use a razor blade instead, just to get cleaner cuts. I have like dozens of these, so we're not going to be using this knife, but thanks. So we have our tray, obviously our pink tray. They did send these really good tweezers. I've been trying to get my hands on these kinds of tweezers. So I have to get rid of some of the. And it's a nice little, chomp. Got a wood poker, which is nice because it's I can use it to point. I was like, what am I going to use to point things out for, once we get started. And then this is amazing. This is going to become a kid's toy afterward. It's a double magnifying glass. Okay. Obviously not useful to anyone here. So that's that. I don't think I'll be using it on this. They gave me a ruler. So that's pretty cool. Ruler. So I'm gonna get my gloves on. Feel free to ask questions as I'm doing things. I can get my fingers in here. Hello. Oh, nitrile gloves are the absolute worst. Okay. And then we have left. Ooh, yeah. Oh, it's gonna be so good. It's been in my refrigerator for like almost 2 weeks now. So, all right, let's crack this baby open. Of course, I already put a hole. Not didn't go all the way down. Okay, good. Smells good. Try not to. Okay. Well, this is a bit weird and tough. Just to get it out of here, I am using a new. Seriously? This thing is in here good. Okay, this is the hardest package I've ever trying to break into. Oh my god. Yeah, that's actually one of the first things I'm going to get into. Make sure that we talk about the distinctions, but also the extreme similarities that exist between humans and sheep, which sheep farmers will be pleased by. I just want to get in it, guys. Can I just get in it? Oh my god. Can we even clip that? Goodness. Okay, well, come on. Gently. There's a piece missing. Something you should eat your lunch. Yeah. Just got some of it. Oh, hey, this is a piece of bone. Piece of bone stuck to the skull. It's pretty cool. It's hard. Oop. Okay, so here I have the brain. It's a little smushed. I wonder if I can give it some definition without damaging it too much. Probably not. It's smelly. Okay. So here's the brain. Yeah. This is the bummer with doing it this way, because I already see some issues with it, but we'll do our best. So we're just looking on the outside. And a couple of things that I want to point out before we start breaking into it. So just to point out a few things. So here we have, this is the cerebrum, or the cortex, the cerebral cortex. Okay. This is this bulbous thing back here. That's the cerebellum. And this extending out of the back is the brainstem, right? So we can call this the hindbrain. If you flip it over, we have the hindbrain. We have the midbrain, although the midbrain is a bit spudgy because, vacuum pressed. They sent me a real good specimen, I'll tell you what. And then the forebrain is pretty much everything that is encapsulated by the cortex. So when we crack this open, we'll be able to look at it a little bit more clearly. How firm is it? It's fairly firm. So what actually the brain feels like inside a person's head is far more like Jell-O than when you see a brain out in the wild, or in a formaldehyde kind of solution. It's probably not formaldehyde, but their secret solution, or whatever it's called. So it is firm. I would say, what's a good word? What's a good word for it? I guess I don't eat anything like that. You know what? It kind of feels like raw meat. And so, no, it would not crumble in my hands. Although the connection of the midbrain to the forebrain, this connection right here is pretty. So if I started flailing it around, you can see that like it kind of falls like that. So you got to be careful not to let that dangle. Now, it's like a raw steak wouldn't crumble. Yeah, exactly. So one of the main differences that you will see if you were to hold, actually, I should say two main differences that you would see. You gonna smell everything, Murphy? Murphy just shut up. His tail just popped in. One of the, obviously, size. That's the first thing I want to point out is your brain is much, much bigger than this. It's about 3 to 5 times bigger than this, depending on how big of a brain you have, right? And head size doesn't matter because, some of us have small heads but really smushed brains. Yeah. One of the other things that you will notice is the smoothness of the cortex, because sheep are super dumb. So they are. Now, there are convolutions on here, which are the sulci and the gyri that you are so familiar with on a human brain. So there's one here, there's one large one, there's some down here. Okay. But if you were to pair this next to a human brain, there would be far more, even vacuum pressed. So, and then the other thing about the cerebellum is that it's all pressed together too. So cortex, smaller, and obviously because the animal is smaller, but also because sheep have small brains compared to their bodies. So that's the next thing that I wanted to point out is that you look at this, you look at the size of this, and you're like, wait a minute. I thought sheep were well sized. And the answer is they are. They're big. They just don't have any small brains. So they have a very small brain compared to their body. Humans, compared to humans who have one of the largest body to brain size ratios. Okay? Which means our brains aren't the biggest. Consider, whales or elephants, but they're also very large animals. So we have some of the, unlike sheep who have big bodies but small brains, so that's another major difference between human brains and sheep brains, is that they got a small one. You know what I'm saying? They got a small one. Cerebellum is a good size compared to the cortex. Oh yeah, that's the other thing that I wanted to say. So when we get into the cortex and we cut open and we see what's going on right here all the way through. Oh, there's another piece of the bone. You'll notice that this portion here looks quite a bit larger compared to the rest of the cortex. And the three-dimensional area, because I don't want to crack it open just yet, the three-dimensional area that I'm talking about here goes from brainstem all the way up and through and then back around. It's almost about half the mass that I have in my hand right now. Compared to a human brain, these areas that I've pointed out, these that I have, this area that I have marked out with my pointer stick is significantly smaller when compared to the rest of the cortex. Here we have like 1-to-1 size of these brain structures, whereas ours are, I don't even know what the ratio would be. It's most like mostly I think it's something, it's got to be 5-to-1 or something like that. And the reason why these brain areas are so much larger is because these are the evolutionarily old brain areas. We're talking about the cerebellum, balance and motor control and motor movements. Brainstem, obviously, this is where the spinal cord goes. The cerebellum is gigantic compared to when you compare it to the size of the cortex, because you've got 4 legs that you need to coordinate, and they're always standing, running, and staying together. You can imagine that you need a pretty large cerebellum to handle all of that coordination with a sheep. Now, human cerebellum is fairly good. Some brain language for you. So, when we talk about the brain and areas of the brain, we have to identify the location. So, looking at the top here, this is called the dorsal side. This is the dorsal side. And that's because, and the best way that I have to remember the dorsal side of the brain is that imagine the animal itself has a dorsal fin. Dorsal fins are the thing that sit on the back of the marine mammal or even fish, right? They all have dorsal fins. And so that's how I remember that's the top. Flipping the brain over, this side, the bottom side, is the ventral side. Now, I don't have a good mnemonic for this one, but I do say that it's just the opposite of dorsal. So if you use the mnemonic for dorsal, like dorsal fin, then you can remember ventral. And that's true of human brains too. Now, because humans walk upright, we actually have to shift that for the spinal cord. But since we don't have a spinal cord, I'm not actually going to tell you the difference there. Just imagine if we were not walking on all fours. What would be dorsal and what would be ventral? And two other directions. We've got the front and the back. So there are two names for the front and the back, depending on what you're talking about. Now, when I talk about a human, I use anterior and posterior because posterior is your bum. So posterior is the back, anterior is the front. But when you're talking about an animal, generally speaking, you use the other two names because they're not cool enough, I guess. They're not cool enough. So instead of anterior, the front is used, the word for the front is rostral. Rostral. And the way that I have to remember rostral is nostril. This is a neuroscientist friend of mine gave me this one. Rostral, nostril. So rostril's the front because that's where your nostrils are, right? And then in the back, instead of posterior, you use caudal. And I don't have a really good mnemonic memory thing for caudal because I just remember that it's the opposite of rostral. Right, rostral, nostril. Oh, okay, the back is caudal. Okay. And then when we make some cuts, I'll tell you the kinds of cuts that I plan on making as we just dissect this. Alrighty. So, let's crack this baby open. Is that what I wanted? Rostral, nostril. Oh! Before I cut it open, which is a bit of a bummer, I'm gonna see if I can get this up. And I just took a chunk out of it. There we go. So these things that I'm pulling up right here, these are, as I'm ripping it, these are the olfactory bulbs. They are giant in comparison to the rest of the brain, and as well as olfactory bulbs in humans. And that's because most mammals have far better smelling capabilities than we do. Far better smelling, right? It also helps that their snouts are so big. They have tons of olfactory receptors in their nose, and then it comes to the olfactory bulb, and they can identify far more scents than we can, and they can identify them from farther away. And there's not much else that I can pull out of I'm gonna take some of this dura matter out of here. Okay. Yep. Take some of that out. So this thin layer that I'm pulling off right here, this thin layer, is the dura matter. It basically keeps the shape of the brain together, keeps all the contents together. Remember I said, maybe Melko is the only one here, but when I said that the brain is kind of like jello inside your skull when you are alive, this dura matter keeps it all together, basically. Okay. And there. And it's all around the brain. And you'll notice it more when I start cutting it open. But inside the dura matter is what's called the meninges. Okay. Brains do not have pain receptors in them. So the closest you can get to pain inside the brain is through the meninges. So whenever you get meningitis, that's inflammation of the meninges. And there you go. Whenever you feel a tension headache, it's technically the blood vessels that surround your brain that are actually paining, right? So the brain itself does not feel pain, which is wild. This is one of the wildest things to me. Additional things to look for in here as I move this closer. Barely, just barely. This is the optic chiasm. This is the crossover point for the optic nerve as it goes to the occipital lobes. Okay, this area right here is the occipital lobe. Down here, temporal lobe, frontal lobe, and then on top, which is not shown at the moment, that's the parietal lobe. So the optic nerves come in from both eyes. They connect here. One side of the retina goes over to the left. The other side of the retina goes over to the right. So in mammalian brains, optic nerves send information from both sides of the retina. They meet in the middle. And then one side of the retina goes to one side of the occipital lobe, the visual receiving area, and the other side of the retina goes to the other side. Okay, so it's fully contralateral. So this side of the brain, if I roll it over here, so if we're looking at the brain like this is the left side and this is the right side. So the right occipital area right here, very back, receives information from the right visual field, and the left occipital area receives information from the left visual field. It's kind of easy to remember, but remember it comes and it joins and then separates back out. So let's do a cross-section. We're going to do a mid-sagittal cross-section, okay? And that's basically going to separate the two hemispheres in long ways. So I'm using my razor blade here. This razor blade is going to probably get thrown away. But using this is going to allow me to cut through the brain, as well as the dura. So here we go. We're getting in there. And I just need to get it deep enough. I wish I kind of had a scalpel, but, I randomly do not have a scalpel. What are you going to do, right? Oh, yeah. Okay. Oh, no. Okay. Hello? There we go. Oh, no, it's more wet. Oh, boy. Come on, get in there. I'm going to separate the, without. There we go. So so here's the cerebellum, where I go into, okay, one final cut. I thought they were separated. One final cut. And then let's do a cross section. I just wanted to separate them. Normally you don't see them separated like that. I don't want to. There we go. They're still connected quite a bit. Okay, let go. There we go. All right, we have our mid-sagittal. We have our two hemispheres. And let's see if I can separate some of this. Now, if this wasn't in their secret sauce, I probably would have tried to fry and eat one of this, but it's probably toxic. So, some of the things that you might be able to see, I'm trying to see which hemisphere is better. So this is only cortex now. So this is clearly the occipital lobe right here. Clearly, he says. Up here, that's the parietal area. Parietal area, generally speaking, comes across mid-cortex. And then the frontal lobe. And then down here is a much smaller, oh no, not a piece. Much smaller temporal lobe. Okay. Because if I put these brains back together, there they are, right? So it's quite a bit easier to tell, if it wasn't so smushed, but it would be quite a bit easier to also tell if it was a human brain. Because the things that I'm pointing out are far bigger. One of the things that I wanted to point out here, let's see, which one has a better, there it is. Yeah. This defined piece that I want to share with you, really the light's in the wrong place, but let's see if we can get it. So this defined area right here, you can see this go all the way back. I'll make a mark with it on this pointer. That's the corpus callosum. That's a thick bundle of nerve axons going back and forth between the two hemispheres. Okay. Get it on there. Is this able to focus? Come on, you can do it. Focus. You can focus. Come on. Focus. Okay. Well, it's not going to focus. So I've made a lot of yeah. The pictures that they sent along, they look far better than what they gave me, but that's all right. So corpus callosum, thick bundle of axons right here, and then right above it is the anterior. Well, it's the singular gyrus. It's a cingulate. Sorry, cingulate gyrus. And it goes all the way around. It's a sort of a curve around the front. Okay, the corpus callosum. Curve around the front of the corpus callosum. And it has my favorite brain area, which is the front part of it, the anterior cingulate cortex, which is probably my favorite, because most of the research that I did in graduate school, some of the biological substrates of the things that I was talking about occur in the ACC there. So, yeah. I'm going to cut out I will cut out the thalamus in just a sec. One thing I wanted to point out here is the cauliflower nature of the Can you? Let's see. Focus. Focus. There we go. A little bit. It's trying. It's struggling. There it goes. Cauliflower. Mmm. So that's the makeup of the cerebellum. And it has a really cool set of neurons. The reason why it gets this shape, this cauliflower shape, and I can kind of spread it out a little bit so you can see it better. The reason why it gets that shape is the kinds of cells that make up the cerebellum. They are tree-like with a ton of dendrites. And they're set up in layers. And that's how you get this tree-like structure, or this cauliflower-like plant structure, I suppose, is really what I want to call it. They're really cool. They're really, really, really cool. And one of the, I would say, ideas surrounding the reason for their structure compared to cortical neurons is that they have so many connections because they're constantly firing, and especially if you're moving, right? So you want to make sure that you have all of the connections. That's why they're so dendrite rich. And that's how you end up with that cauliflower. As I've massaged the cauliflower, you should be able to see it now a little bit better. So you technically do have a right and left. Technically do have a right and left cerebellum. There is a center area, which is generally speaking, or a connection area right here, where the two sides will connect and speak with each other. Okay. You can just like break this open. By the way, I want to thank the sheep who perished to allow me to do this, because this is a real sheep brain. Okay. And the other thing that I wanted to note about the cerebellum is that if I cut some of the dura here, and you can start getting your cauliflower shape here. Cauliflower brain. Yeah, cauliflower brain. Can just squeeze it. So you get your cauliflower there. It looks good. Looks good. Now if I put them back together, can we put them back together? So I wanted to put this back together because I wanted to show just very briefly a couple of aspects of the mid and hindbrain here. So this brainstem is essentially going to the spinal cord. Essentially going to the spinal cord, right? But there's a bit of a bulge right here. That's your pons. Okay, so right here. Your pons. So the cool thing about pons is that, and this specifically refers to dreaming, and I love talking about this. So there is a leading theory out there about the role the pons plays in dreaming, and specifically dreaming during REM, rapid eye movement sleep. So the theory goes that when you're awake, the pons is allowing information from your brain to travel down your brainstem. Just beep boop boop beep, beep boop boop beep, beep boop boop beep. And you may be familiar with how when you are in rapid eye movement sleep, you are technically supposed to be paralyzed. You're technically supposed to be paralyzed. Now, there is REM behavior disorder where you don't become paralyzed during REM. You don't. And so you actually can start actually acting out your dreams. Very technical beeps and boops, yes. So the pons is its job is to stop information from traveling down into the brainstem and the spinal cord while you are dreaming, while you go through rapid eye sleep. Okay? So while you're dreaming and in REM, the pons says, no, you can't. You're not allowed to go through here. You cannot go through here. Further up, which is undefined on my smushed brain here, is the medulla. So it's about right here. We'll call it that just because it's not really defined on my brain because it's smushed. In this area here, the medulla with concussions is because, let me put my brain together here. Oh. So here we have a brain, and here it's going to meet in an immovable surface. Now, because it is jelly, it's going to come, because it's under its own, it's traveling at its own speed, right? And it comes up, my palm being the front of my skull, right? So let's just say I'm hitting the front of my head, my forehead, let's just say, right? Or I'm doing a header in soccer, okay? That smushes my brain. And then it has to bounce back. And one of the things that occurs with the jelly brain is that it sloshes backwards against the back of your skull and gets pushed. And then it does that, and it goes back and forth, back and forth, back and forth. And this, the cortex and all inside of it, is basically getting accordioned. And that leads to shearing along the cortex's top, right? And that shearing basically breaks brain cells, right? So the harder the impact, the worse the concussion is, right? One of the reasons why it can do that is because it is more gelatin in your skull when you are alive. Because you got to remember that it's also surrounded, it's encapsulated by cerebrospinal fluid. So your brain's basically sitting in water and nutrients and salt and things like that, which makes it just gooey for the most part. And then once you take that away, it starts to harden, right? Which brings me to the next part of the discussion here. I am going to do what's called a coronal cross-section, which is not as nice as it sounds. And I don't really have a good mnemonic. We did a mid-sagittal cut, and now what I'm going to do is a coronal cut. And really the only thing that I can think of as a coronal cut is it's a straight down cut. It's a straight down cut. So I'm just going to do a straight down cut just like that. Went through the dura, down in there. Whee! So this brain's never getting put back together now. Ooh, yeah, look at that. Look at that. That's really cool. Is there a correlation between intelligence and brain size? Not with intelligence and brain size. The correlation, generally speaking, comes from body to brain size ratio. So humans have the largest brain size to body ratio, but we don't have the largest brain. Chimpanzees and gorillas have larger brains than we do. Whales and elephants have larger brains than we do, but they also have larger bodies than we do. So when you compare intelligence, you want to look at two things, in my opinion. You want to look at the smoothness of the cortex. So how many convolutions, how many sulci there are. Sulci are the valleys versus gyri. Gyri are the mounds, the hills, if you were. So a sulcus and a gyrus. This is actually the center sulcus, I believe. So that's the first thing to look at. So you look at smoothness. The human brain has far more convolutions than any other animal that exists, and mammals obviously have more convolutions than any other type of animal, like lizards, birds, or I should say reptiles. And so that's smoothness of the brain. And then the second thing is what I've already said, which is the body to brain size ratio. What is that? So I cut open. Which piece is better? Which piece is better? Oh, that's my mic. Okay. So one of the remaining things in this dissection that I wanted to point out is you can sort of see the difference here in coloring, right? So we have like a pattern here coming from this stem. There's a pattern right here. There's a pattern that goes up here and here and here. This is the white matter, and then surrounding it, which is the darker color, is gray matter. So we have white matter and we have gray matter, and this is going to go. This is going to go into a discussion that happened a few minutes ago with like the molecular constituencies of kind of molecules that are in the brain. So white matter is so named because it looks white. It's got a lighter shade of beige to it, we'll say. And the darker is the gray matter. And it's called white matter because of a lipid sheath. Oops, dropped it. And that lipid sheath is called myelin. Myelin. Now, myelin coats axons and it speeds up an action potential. So myelin speeds up neuronal chats. So this white matter right here is a bunch of myelinated axons. And in the gray matter, we have cell bodies and unmyelinated axons. So their transmission is a little bit slower than this myelinated stuff right here. Okay. Now, the reason why myelinated, this is blue thing in here. Myelinated neurons are faster because fat cells are not good conductors. Okay. And an action potential is a bioelectrical signal. Okay. And in order for the action potential to pass along the axon, it has to jump from node to node along the myelin. So it's kind of like kind of goes like this. And those nodes are called the nodes of Ranvier. And that's a good question, Rambler. Why not all myelinated? Well, it takes significant amounts of energy and resources to produce a myelinated axon. And it is not as regenerative as you think it is. So you're probably familiar with multiple sclerosis, right? Multiple sclerosis is 100%, which is why the symptoms of multiple sclerosis are so varied. And that's because myelinated neurons are being attacked. Okay. And the myelin sheaths are being essentially destroyed from those axons. And so that's going to impact cell transmission, which is why you see the kinds of detriments that you see with somebody who is suffering from MS, right? So humans have a lot of white matter. You can see the sheep doesn't really have a ton of white matter. And the only way that you're going to actually see it is by doing this coronal cross-section, because you're not really going to see white matter along this, along the mid-sagittal section here. That's because this is technically surface of the cortex, and on the surface of the cortex is gray matter. Yeah, humans have a lot of white matter compared to other mammals. So it's kind of hard to tell in this one here, but there's a lot of gray matter. Let's squeeze this in here. No, I can't really. Into this gyrus. The dura is pretty hefty on this animal. Trying to separate this gyrus or the sulcus, these gyri from each other. It's not a lot. It's not a lot. Considering the size of their brain is already so small, this isn't a lot either. Let's do another. Let's do a horizontal or transverse cut down the center. Well, kind of center-ish. I don't want to do it too hard because cut my finger open. Good old transverse cut. So this is the ventral portion of the cut that I just did. Okay. And you can kind of see the mapping here of white matter in this section. So if we were looking at it like this, I just basically cut a good portion of the temporoparietal junction area right here. That's what this area would be right here. The temporal lobe on a sheep is not defined well enough. So I can't really tell, but there you go. So we've got so this is the frontal lobe right here. So if we look at it like this, we've got some white matter in the frontal lobe here. This is the sheep's problem solving, simple problem solving. Hey, maybe we should, I don't know, form a group so that that wolf can't get us. And then coming down in here, and then back here is the occipital area, and you can see connections here. This is the visual area. Okay. Visual area of the brain. Yeah. Good stuff. Any surface is going to be gray matter. And then the inside is going to be the white matter. Okay. So if I were to break apart the convolutions like I did right here. So here, I'm putting the um, stick here in a sulcus. And so here's a gyrus, right? Right here. Here's a gyrus. This is 100% gray matter at the gyrus part of it. As you go into the sulcus, so where I have it right here, that's where you see the white matter. That's where you see the changing of the guard there. So if I were to do that on every single convolution that I see here, you would notice the difference. All right. That's as much brain as I'm going to touch.